Very good morning, students. We'll start our lecture 7 of Fundamentals of Agronomy course with seed and sowing. What is seed? We call seed as a fertilized ovule. It is a fertilized ovule consisting of intact embryo. It consisting of an intact embryo, stored food material that is endosperm, and seed coat which has viable and has a got capacity to germinate, and the embryo is divided into plumule and radicle. The plumule is the shoot part and the radicle is the root part. A seed is a fertilized ovule. It is a fertilized ovule, not ovary. It is a fertilized ovule consisting of two parts, that mainly the embryo. Embryo is divided into the plumule, that is the shoot part, and the radicle, which is the root part. It has a cotyledon and that outer seed coat which has testa and tegmen. Just testa and tegmen. So if you see an endosperm of a seed, if you see a seed, this is an example of a maize seed. It has a seed coat, it has a cotyledon, and it has a coleoptile. Coleoptile, that is a plumule sheath. It is a plumule sheath. Coleoptile, which is a plumule. And the plumule is where the first leaf emerges, and the radical part which gives you the root. And we have coleorhiza, that is the root bearing structure, root. Coleorhiza. This is a seed which is covered with an seed coat. It has stored food material. We call it as endosperm. It has the cotyledon, from which the plumule, and the next coleorhiza, from which the radicle emerges. This is the stages of the development. The seed where the first root emerges, followed by shoot emerges, that gives to a coleoptile, then formation of roots, the prop roots, the main or brace roots, and the final plant. See, the plant population is the multiplication of the plant either by, plant propagation is the multiplication of plant either by sexual means and asexual means. Sexual means by seed and asexual means by stem cutting in case of sugarcane. With respect to rooted slip, a good example is tuber, with the tuberous roots or corms, rhizomes, grafting, budding, layering, and in a case of horticulture crop. So the propagation of the plant material can be done in both sexual way and asexual way. When it is a sexual way, it is through seeds. When it is an asexual way, we use various non-fertilized techniques, such as stem cutting, root slips, tuberous crops, corms, grafting, layering, mainly in horticulture crops. And forage grasses such as napier, guinea grass, paragrass, mainly propagated through the stem cuttings or rooted slips. Then after understanding the seed, we need to differentiate between the seed and grain. What is the seed? Seed is a viable and used for sowing. Seed is viable and it is used for sowing. Whereas grain seeds are for human consumption. It may be viable or may not be viable. It is mainly used for human consumption, whereas seed, it is used for the sowing. It should be viable, need not to be viable. It should have maximum genetic and physical purity. No such purity concern is there with respect to the grain. Should satisfy minimum seed certification standards, that is at least 99% of germination percentage, purity percentage. Such standards should be met with respect to seed. No such requirement with respect to grain. It should be completely treated with the pesticide, fungicide to protect the seed against the stored pest and this fungi. Whereas no such chemical treatment is with respect to the grain. Should be compulsorily certified with respect to the seed, but no such conditions are with respect to grain, and it should satisfy all quality norm, and there is no such quality norm with respect to the grain. So what is the characteristics of a good seed? How you consider a seed as a good seed? The first is, as you say, reap as you sow. So whatever the outcome you get is based on what you have sown. So a quality of a seed, it should be pure. It means that there should be no adulterant or no dust material. It should have higher germination percentage, 99 percentage, and good vigor and viability. Its seed should be free from the seed-borne diseases. The seed should remain in the optimum size and shape, and it should have optimum color, that is such as maize yellow color, rice brown with seed coat, husk. So it should have optimum color. Seed should be free from weed seeds and inert material. This is very important. Seed, for example, mustard seed, when it is mixed with the tura seed, it is not. It is very difficult to differentiate. But the tura is poisonous to the human beings. For example, seed should be free from the insect, mainly rice, grain moth, or even pulse beetle. So such are the major store grain pests. It should be free from the insect. It should not be broken. It should not be containing any required. It should be containing a required amount of moisture. For example, for oil seeds, the storage moisture content 8 percentage. For cereals, at least 12 percentage of moisture content. So such moisture should be there, and it should be in a proper stage. The viability should be there. It should not be of a greater long stored duration. It should be germinate rapidly and produce vigorous seedling. It should be fresh and clean, and it should be plump. It should be large and plump, not dented kind of thing. So after understanding the type and characteristics of seeds, we have classified the seeds into four classes categories based on the genetic purity and the stage of development. The first is the breeder seeds. Breeder seeds, as also called as nucleus seed, it is developed or produced under the supervision of a plant breeder, and these are very lesser quantity, and the purity percentage of breeder seed should be 100%. It is stored in a golden yellow color tag, with a golden yellow color tag. The next is foundation seeds. It is have a bag white color tag. It has a purity of 99.9%. And the progeny, it is a progeny of a breeder seed. Next to breeder seed, it is a foundation seed produced from a recognized seed producing agencies or a public or private sector under supervision of seed certification agencies, and the color is white color tag. The next certified seed, the progeny of foundation seeds, and it is registered seed grower under the supervision of seed certification agencies, which is stored with a blue color certification tag. Then truthful label seeds. This is a progeny of certified seeds which has opal green color tag. Opal green color tag. Remember the tag and purity percentage. It is around 95% purity. This is 99.5% of germination percentage and purity. So foundation breeder seeds 100% purity percentage. Foundation seeds 99%, certified seed 99.5%, and truthful label seeds it's a 90%. With respect to tag, it is golden yellow color, foundation seeds white color, certified seeds that is azure blue color, then truthful label seeds it is opal green color. These are the tags of the particular bank. Then after understanding the types of seed or categories of seeds based on their production technology, further we need to understand the process after we go for sowing of the seeds. This is a well viable seeds. When I go for sowing in the soil, what happens? It germinates. How it germinates? Where it absorbs the water, it imbibes the water, then it results in activation of certain metabolic reactions, metabolic enzymes in the seeds. So with that activation, the coleorhiza and coleoptile emerges from the seed. The seed germination is a process by which a seed embryo develops into the seedling. It involves reaction of metabolic pathway that leads to the growth and emergence of a radical, seed root, and a plumule or a shoot. The next is seed viability. What is viability? It is ability of a seed to germinate. We call it as viability. That is, embryo to germinate, we call it as viability. Then we have two kinds of germination. The epigeal germination and the hypogeal germination. Very important with respect to exam point of view. Epigeal germination in dicots. or in pulses, the cotyledons are brought out of the soil, occurred elongation of the hypocotyle, and this type of germination where cotyledon emerges out from the seed, we call it as apigeal germination. If you have studied, if you have noticed the crops such as pulses, green gram, black gram, where the seed cotyledon emerges out from the soil into two cotyledon. Such type of emergence we call it as epigelial germination, where hypocotyle extension takes place. Whereas hypogeal germination mainly seen in the cereals where cotyledon remains inside the soil and most plumule growth is pushed upward by elongation of epicotyle. So we understand the difference between the apigeal germination and hypogeal germination. If you see here, what is happening here? A seed with its hypocotyle is coming out. It is coming out and it giving where cotyledons are above the soil surface, we consider it as a apigeal germination. What is hypogeal germination? This is a seed, consider cereal seeds, maize, where its hypocotyle is inside the soil itself, where the cotyledon are inside the soil, only the epicotyle comes out with the leaf, whereas the cotyledon remains inside the soil, but here the cotyledon is above the soil. Such type of germination we call it as epigeal, and that other is the hypogeal. Epi means above, the gl means earth, where hypo means below, gl means earth. The seed cotyledon emerge out of the soil, the cotyledon remains inside the soil, cotyledon turn into green and acts as the first leaf. This is very important. Here the cotyledon turn into green and acts as the first leaves for the photosynthesis. Cotyledon play no role in case of photosynthesis with respect to the hypogeal germination. The hypocotyle raises to the higher length and the epicotyle raises to the higher height. The terminal portion of hypocotyle is curved to protect the plumule from the friction of the soil. The terminal portion of the hypocotyle is curved to protect the plumule from the friction of the soil. The terminal portion of the hypocotyle is curved in order to prevent the epicotyle from the friction of the soil. Then energy for the growth of family derived from the cotyledon. Some pulses are example of epigeal. The terminal part of epicotyle is curved to protect the plumule from the friction of the soil. Epicotyle, here it is hypocotyle. Hypocotyle is the upper part, epicotyle is the below part. Here epicotyle is curved so that it should not, in order to overcome the friction from the soil. See here a good example of epigeal germination and hypogeal germination, a pea seed and a bean seed. Pigeon pea are example of hypogeal germination in case of pulses, some exceptional. Then after understanding the types of germination, we shall understand the factor that affects the germination of the plant. The first is the external factors that affect the germination of the plant, such as moisture, temperature, oxygen, light and substratum. Moisture, generally higher the moisture, that results in poor germination of the soil. So ideal moisture condition is required for the germination of the seed. Then temperature, optimum temperature such as around close to 20 to 25 degrees required for the seed germination. You see here, we consider it as a cardial temperature. Then oxygen, proper respiration and physiological function has to be taken into the soil, so proper aeration should be met. Then light requirement, it is not considered essential, however few seeds are photoblastic which germinate in the presence of light, so optimum light availability is required for the growth of the plant. Then substratum, it is a medium used for the germination. In laboratory you use certain media for germination or paper towel method, a soil as an ideal soil medium for the germination. These are the external factors that determines the germination of the seed, such as temperature, the soil moisture, the oxygen, the light and the substratum. And the internal factor that decides the germination of the seed, such as the first growth metabolic factors or food or axines Auxin is a derivative of amino acid. The zinc plays a very important role in availability of auxin. So food and auxins, embryo feeds on the stored food material and young seedlings prepare its own food, and auxins are the growth promoter, hence quite essential for the germination. The viability, that is ability of a seed to germinate in a very fast manner, it is viability. That also determines the internal factor that determines the germination of the seed. And dormancy, it is a failure of mature viable seed to germinate under the favorable conditions. Few seeds such as groundnut, where they have innate dormancy that does not allow the seed to germinate. So such factor, the internal factor, also decides the germination of the seeds. So we were discussing about the factor affecting the germination. The two factor, external factor and internal factor. External factors such as moisture, temperature, light, substratum, and even the conditions. So whereas the internal factors such as the viability of the seed, the food, stored food material, the growth factors such as auxins, or even the dormancy of the seeds, that determines the germination of the seeds. Then what is dormancy? Yes, the next breaking of dormancy will start with the next class. Hope you enjoyed the class. Thank you.